right, well, we are the hardy ones today. Looks like not a whole lot of you wanted to venture out into the cold. That's kind of understandable. I'm hoping that I have my audio issues worked out. Hopefully it sounds a little better. I'm not getting the feedback now quite so much, a little bit of an echo. Just tell me if I stray too far from that microphone, let me know if that impacts whether you can hear me or not, but it's good. Most of you are up towards the front, so that's good. All right, so a few announcements. Of course, our first exam is Wednesday, and I'm going to assume, unless I've heard from you or hear from you very shortly, that you're planning on taking the exam here in the room in person. There will be 63 questions. They are all multiple choice. Let's see, just a second. There we go. So 63 multiple choice questions. So you have 63 questions in 75 minutes. So you have more than a minute to take to answer each question. I don't anticipate that you'll really be crunched for time. So you shouldn't be rushing through that. I typically will start handing them out a few minutes before 1:30. Takes me a few minutes to get around to everybody, but I tried handing them out as people came into the room, and it was just too much of a bottleneck last semester. So we'll get them all out, and again, you shouldn't have to rush through that. Again, contact me if you really, only people, special circumstances am I allowing people to take it by respondus, people that are, have medical conditions or living at home, something like that. So talk to me if that's you. I'm giving it to a few people in a smaller group setting. So again, if you have concerns, talk to me about that. But most of you will want it taken here. All right. Also remember, your homework is due at 1:30 on Wednesday. So I've seen some of you haven't started yet. It's time to get started. So it's not such a burden for you. I have it set right now not to give any credit if it's late. So, if you have some special circumstance come up, don't be afraid to talk to me about that. But again, 1:30. It was brought to my attention, especially in chapter 12, there's a number of questions that in regard to fungal reproduction, which we didn't cover this semester. So I went in and I got rid of those questions because there were quite a few. So my apologies if you already answered them and had to go through that strife. I realized what I did earlier in the semester. I went through and I got rid of those questions, but I forgot to hit save changes. So that was my bad. I'll try to be kind of more aware and proactive at looking at assignments and making sure I don't ask you about a bunch of things that you're not responsible for this semester. So, all right. So if you have questions about the exam, don't be afraid to, catch me afterwards or email me. I did want to just show a little support for the Microbiology Club. They're having their first meeting of the year tomorrow night in person over at Kirchhoff. Also possibility to do that by Zoom if you're interested. It's more of a kind of let's meet each other and decide what we want to do this semester kind of thing. So if you're interested in that, just wanted to give you that information. All right, so what we covered today will not be covered on Wednesday's exam. I will just send out an announcement to just make that ultra clear, but hopefully that information gets through. Sorry, 1 second. Let me let one more person in here. It's a little cumbersome having a lot of people in by Zoom, but I'm willing to try to make that accommodation. Okay, so again, not covered on Wednesday's exam. We are going to kind of fly through a lot of this. So some people just dread metabolism. The way that we are covering it in this class, of course, we were interested in metabolism that is carried out by bacteria, especially. So you've had metabolism in other classes that may have been focused more on human metabolism. A lot of it is the same. So whatever you can remember from other classes, certainly that's going to help you. We are not going through metabolism in the depth that you would get in a biochemistry class. We're not memorizing a bunch of pathways or a bunch of enzymes or structures. We're looking at it more from a kind of big picture point of view. And we're especially interested in the things that are unique to bacteria. So we make comparisons, but mostly, again, from my microbial point of view. So we're going to start out just today by reviewing the components of metabolism. So the kinds of things that you kind of have to understand to look at a pathway, like redox reactions and electron carriers, enzymes, a little terminology there. I think we will probably get pretty close to getting through the pathways of aerobic respiration today. So that's glycolysis, some alternatives that some organisms have to glycolytic pathways, as well as the Krebs cycle and electron transport chain. Okay, and then probably next time we'll move more into anaerobic respiration, which of course is something that we don't do, as well as fermentation, and try to highlight the differences between those two processes. Okay, all right, don't be afraid to jump in if you have a question. I know it's a little bit hard in the environment that we're in, but don't be afraid to ask. All right, so we have to talk about metabolism because, of course, this is how organisms gain ATP, gain energy. And of course, we just got done talking about kind of describing growth of organisms. And of course, they can't grow and reproduce if they don't have energy to do that. So maybe we're doing that a little bit backwards. But so when we're talking about the metabolism in a cell, that just means, all the biochemical reactions of a cell. And those reactions, we can label those as either anabolic or catabolic. So remember, anabolic are reactions that build molecules. We could also describe them as endergonic, meaning that energy is required for anabolic reactions to take place. And then the reverse of that, catabolic reactions, these are reactions that break down molecules. Okay, and of course, in catabolic reactions, we get the release of energy instead of energy inputted. All right, so the graphic from your book is kind of nice here. Kind of reminds us that the anabolic and the catabolic reactions within a cell, they have to be balanced. You can't have, a bunch of energy being spent without energy being generated. All right, so in a catabolic reaction, so a cell might absorb some nutrients here, break those nutrients down into some kind of precursor molecule. So say, this is a bigger sugar that's broken down into smaller sugars, for example. Those reactions are not going to be 100% efficient. Okay, some energy is going to be lost as heat. Of course, your car engine, kind of same idea. It's not very efficient, so you get a lot of heat generated. Okay, that's why we have, a body temperature, because we have heat released as part of these byproducts of these reactions. And then anabolic reactions are going to take those precursor molecules and build into, larger building blocks, macromolecules, things that the cell needs, for its structure. Cellular processes, maybe storing energy in some cases. All right, so. All right. Another thing that is obviously kind of the confusing concept of oxidation and reduction reactions. We often call those redox reactions. We take a little bit of one word and a little bit of another word to make a different word. So redox. There's a nice little YouTube video here that's kind of old now. It's from Crash Course Chemistry. It's like 10 minutes long. If you just watch like the first 2 or 3 minutes, it's quite interesting. He talks about where that name redox comes from and how it's not a great way to describe really what's going on anymore. But one thing that he is fond of saying in that clip is that it's all about the electrons. So it's really important to understand where the electrons are going. Those electrons are going to get converted into lots and lots of ATP during aerobic respiration, especially. So it's important to follow the electrons. So we kind of have to understand what we mean by oxidation or reduction. So many of you have learned the acronym oil rig. Okay, oxidation is loss of electrons. All right, so here we have an electron donor. It's losing its electrons, so we say it's been oxidized. Here we have whatever, a molecule that's accepting electron. When it gains an electron, we say it's been reduced. So that always, didn't sit well with me. Why did we say when it gains something, it's been reduced? Well, you can think about it as the net positive charge has been reduced. Okay, because it's gaining something that's more negative. So that may be helpful. I think something else that is always confusing about this is that we forget, because we don't write this in, that protons often accompany electrons. Okay, so we write the protons in the equations, but then we don't write in the electrons. Okay, so whenever you see a proton, you assume that there's an electron with it. Okay, so a hydrogen ion, that's a proton and an electron. Okay, so proton and electron. So whenever you see a proton, you just kind of assume that there's an electron traveling with that. Right, so that hopefully is illustrated here as we talk about our electron carriers. Okay, we can't just let electrons kind of go wherever they want in a cell, because electrons, when they're unchaperoned, cause a lot of cellular damage. They can, pull, other electrons off of other molecules and cause damage. It's like, high school. You don't have a high school dance. You don't have a bunch of teenagers, and you just say, "Okay, go have a dance." Or you have adults there to make sure things don't get out of hand. So that's kind of the way you can think about these electron carriers. Okay, they're chaperones for these electrons. Another way you could think about them, the NAD, the FAD, NADP, is that they're empty baskets. And when they have electrons in them, they've got something in their basket, and they dump it out somewhere, and they go back and fill up their basket again. So they're kind of just alternating between a full basket and an empty basket. Okay, and that nicotinamide adenine dinucleotide, NAD, that actually carries a hydride ion. So when NAD accepts these electrons, it can carry 2 electrons and a proton. So its reduced form over here, this is the oxidized form. When it's reduced, it's NADH plus, and then plus another proton. Okay, so in other words, let's change here. The NADH is carrying a hydride ion, which is 2 electrons and a proton. Okay. So that's why it's the NADH, and then there's this proton left over. The protons, they don't have to be chaperoned. They don't cause the same kind of damage. Okay, FAD can actually carry a hydrogen atom. So it's, whoops, F, there we go. In its reduced state, it actually FADH2. And then NADP also carries a hydride ion. So just remember, there's actually 2 electrons associated there. Oops, 2. We just don't write those in. Okay, and the other thing that I'll mention about these different carriers is the NAD and the FAD are carrying, NADH, FADH2, are going to carry their electrons to the electron transport chain, at least during respiration. Okay, and we'll talk about the difference between respiration and fermentation shortly. Okay, the NADP, these electrons don't get carried to the electron transport chain. They are actually used to power anabolic reactions. Okay, biosynthetic reactions. All right, so they kind of have a different place that they transport their electrons. All right, so of course, the energy currency of the cell, that's ATP. And this is a molecule that recycles between its energetic form with the 3 phosphates. It's got these bonds that are high energy bonds here between the phosphates, of course, and it recycles between ATP with 3 phosphates and with only 2 phosphates, ADP. Okay, how is phosphate added to ADP to make ATP? We have 3 ways that we describe how that phosphorylation, that addition of a phosphate, takes place. Okay, and there's substrate level phosphorylation. And oxidative phosphorylation. And the third way is through sunlight, so photophosphorylation. We are particularly interested in bacteria that cause disease. There are no photosynthetic bacteria that cause disease, so we're not really going to worry too much about the photosynthesis and photophosphorylation. Oh, photophosphorylation. Yeah, I didn't spell that right. Too many syllables. Okay, so let's look at an example of what's the difference basically between substrate level phosphorylation and oxidative phosphorylation. We're going to pick glycolysis apart a little bit more in just a minute, but just use kind of the pathway as this is an example of substrate phosphorylation, or within this pathway, there's a couple times that this takes place. Okay, so we can define it. The definition's a little bit clunky, but the transfer of phosphate from ADP, a transfer of phosphate to ADP from another phosphorylated organic compound. So the substrate here is the source of the phosphate. So in glycolysis, for example, with this compound right here, you can see the phosphates on it. Basically, the phosphate is ripped off that substrate and added to ADP to make ATP. Okay, so this is just an example of a couple of times where when ATP is made, it's by substrate level phosphorylation. So basically, this takes place during glycolysis and the Krebs cycle. Okay, that's when we get substrate level phosphorylations. Okay. Oxidative phosphorylation is a little bit harder to explain. It's not just that there's oxygen involved. Okay, so that's kind of a misnomer there. Oxidative phosphorylation is going to take place at electron transport chains. And again, we're going to go through that pretty carefully, but it's because the NADH, those electrons that the NADH is carrying, this gets oxidized back to NAD. And those electrons kind of pass through the electron transport chain, and some electron acceptor, it might be oxygen, it might be something else, eventually picks up those electrons. So it's because of this oxidation here that we call this oxidative phosphorylation. It's a result of that oxidation that ATP eventually gets generated, again, at the electron transport chains. And this is always going to take place in some kind of membrane. So I posted this little graphic here as kind of a reminder, if the dam is kind of an analogy for the cell membrane. And actually during the oxidative phosphorylation, we get lots of potential energy out here. The lake, that's lots of potential energy. When that flows through the turbines of the dam, we get electricity made. It's kind of the same idea, electron transport chains. You have lots of protons outside, they get pumped outside of the cell. And then when they're allowed to flow back through, then we get the generation of ATP, the phosphorylation of ADP to ATP. All right. So more about that later. Again, photophosphorylation. We're not going to worry about photosynthesis because we are, again, more interested in organisms that live in our body. So skip the whole photosynthesis in this chapter. Okay, so a little review on enzymes. You know, some of you, this is, you know, of course, kind of things, and I find other students struggle with the terminology here. So remember that enzymes, they work on substrates. We always draw whatever enzyme we're talking about, we draw it over the arrow and turn those substrates into some product. Okay, so enzymes, they decrease the activation energy. So these are reactions that would take place in the cell, but so slowly that life wouldn't be able to be maintained. So enzymes speed up those reactions. Here, instead of substrates, they say reactants, and you end up, again, with products, okay? Okay, so some enzymes are protein only. Okay, all enzymes have a protein component. And of course, they usually often end in ase. So if you see a word like lipase or protease, you know that it's an enzyme. Not all enzymes end in ase, but some of them do. Okay, so some enzymes just are composed of a protein. We call those simple enzymes. But a lot of enzymes have a non-protein component. Okay, so if you take a look at this figure from your book here, some enzymes will need some inorganic part that we would call a cofactor in order for the enzyme to function. And when we talked about what kind of nutrients cells need, we talked about both some macronutrients and micronutrients that might be necessary just for enzyme function. So things like copper or magnesium, iron. Those are all things that often function as enzyme cofactors. Something just so the chemistry of the enzyme will work. If that cofactor is organic, then we refer to it as a coenzyme. So there could be, both a inorganic and something organic that's necessary, not a protein that's necessary for the enzyme to function. Okay, so the protein part is called an apoenzyme, and the whole thing all together we would refer to as the holoenzyme. So a little bit of terminology there. Of course, the active site, that's where the substrate binds. Okay, so you've all heard the lock and key analogy. The substrate binds into the lock, fits just right, and then the substrate is changed into something else. Okay, so here's an example. I didn't post this, but this is just a figure from your book. Just an example of the function of an enzyme. So here's the enzyme here in blue. It's acting on a substrate like fructose 1,6-bisphosphate. The enzyme, in this case, what does it do to the substrate? It splits it. Okay, it breaks some bonds and to break that substrate into two different molecules. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That's the lysis step in glycolysis. All right, so then the enzyme cycles back and acts on a different substrate. So the enzyme is not changed. Okay, but the substrate is. Okay. And of course, enzymes can have different activity rates depending on the environment that they're in. So just like cells can grow fast or slow depending on their environment, same thing with enzymes. And those two things are actually really closely coupled together. If lots of the enzymes in a cell function best at around 37 degrees, that's probably the ideal growth temperature for a cell. So that makes sense. You get a little hotter and those enzymes denature and their shape changes and they're not going to function. pH, there's often some ideal pH that the enzymes will function. It might function a little bit more slowly at a different pH, but they're going to have an ideal pH. And an enzyme is going to work most quickly if there's a certain concentration of substrate to a certain extent. So if you have, a constant number of enzymes, you get more and more substrate, you get more and more activity. But if you get, more substrate than enzyme, it's not going to work any quicker, obviously. So you can change the enzyme concentration or substrate concentration. Okay, and a lot of that regulation we're going to talk about when we get into our genetics unit. Sometimes it's best to regulate whether the enzyme actually gets made or not. But there's other ways to kind of dial in just the right amount of enzyme activity to be, kind of most efficient for the cell. Okay, so there's different ways that an enzyme can be inhibited. Competitive inhibition, that just means some molecule is going to block the enzyme's active site. Okay, so if you look at, in this example, the substrate that fits into the active site of the enzyme, anything that's a competitive inhibitor that would fit in that same active site, you can see that its shape is very similar to the substrate. Has to be in order to fit into that active site. Okay, so if there's only a little bit of that inhibitor, then most of the time the substrate will fit in there instead of the inhibitor. But it depends on, again, the concentration of that inhibitor as to how much that enzyme will be inhibited. And then, of course, you have non-competitive inhibition where there's some other site that inhibitor binds. So allosteric, really you could think of that as something other than the active site. Okay, so when that inhibitor binds, say over here, that changes the formation of the active site. It's distorted so that the substrate doesn't bind anymore. Really, a better way to think about this is non-competitive or allosteric control rather than just inhibition, because sometimes we might have an allosteric activation where the enzyme needs something to bind at an allosteric site in order to change the formation of that active site so that the substrate now can bind. It can only bind when that activator is present. Okay, so there's different kinds of control, not just inhibition. So an example of allosteric control is in maybe some kind of pathway that a cell will have, where it's a way to kind of regulate Obviously, if a cell has too much of something, it doesn't want to keep making that. So feedback inhibition. An example here might be a bacterial cell that a lot of bacteria can make their own amino acids. So if there's a pathway, it's like 5 different enzymes for the bacteria to make its own tryptophan. And if there's a lot of tryptophan in the medium, that tryptophan, say this is tryptophan here, can actually bind back up to the original enzyme in some allosteric site and change the formation of the active site so that basically the pathway will stop. So again, you don't want, if you have a whole bunch of it, it doesn't make any sense to make more. Sometimes we might grow bacteria in a medium that already has tryptophan in it, so then it doesn't want to just keep making more and more. And that's, I can't think of an example where this would happen as a competitive inhibition, where that end product would fit, you know, right into the active site. That would mean it would be very similar to the substrate itself. So, and most examples here are an allosteric kind of feedback inhibition. All right, so that's our little review of kind of the things that we need to kind of all put together to look at these pathways. Again, I try to give you a good idea in your learning objectives as well as to what kind of things that you're going to need to know. We're going to pay particular attention in these pathways. How much ATP has to be put in? How much do we get out? There's some key juncture points that we have to understand. Well, when would a pathway likely go in this direction? When would it likely go in another direction? We're going to look at the pathways themselves, paying particular attention, again, to the products. What do you start with? What do you end with? What do you get out of it? Okay, we're not going to worry about naming all the intermediates or drawing structures or the enzymes that are necessary for the cell to have. Sometimes it's helpful to kind of count the carbons, right? So if you start with a 6-carbon glucose, oh, you're ending with 2 3-carbon pyruvates. Okay, so you still got the same amount of carbon. If you don't, where did it go? And how did we lose it? So sometimes that's helpful. And like I said before, follow the electrons, because those electrons are in respiration what makes us end up with lots of ATP. That's the really important thing. So here's the kind of overall diagram that I'll refer to a couple times in your book. It's kind of a nice overview, a summary of glucose catabolism. We always kind of talk about metabolism and we start out with glucose. Most cells are pretty flexible in that they can use lots of other things for energy besides glucose. But glucose is kind of the most readily available thing that a cell would like use first if it had the choice. Okay, most cells can use proteins if there's no source of sugar. They can use lipids if nothing else is available. So we'll talk about that kind of briefly, what happens if proteins are the only thing available or lipids are the only thing available. But again, they always are going to choose glucose first, almost always. Okay. And I like this diagram because it kind of course, on the side that's green, and if you made copies, this isn't clear, but the green side is showing the respiration pathways, and the orange side is showing the fermentative pathways. Okay, usually an organism is going to metabolize by some respiration pathway whenever possible, because they get more ATP, more energy from utilizing respiration than fermentation. But it all comes down to whether there is a final electron acceptor available. If there's not, then the cell doesn't have any choice except to use some fermentative pathway. Okay, because if they don't have any, they have to have somewhere to dump off these electrons that they're getting out of glycolysis. If there's nowhere to dump those off, there's no suitable electron acceptor, then even glycolysis will stop, because we have to have NAD cycling back to pick up electrons to keep that oxidation of glucose going. So that can be accomplished through some fermentative pathway. You don't usually get any more energy out of a fermentative pathway, but at least they get the ATP a little bit from glucose. Okay, so we're going to talk through the glycolysis pathway. There are a couple of alternatives to glycolysis that we're going to talk about as well. And again, glycolysis is, whether a cell does fermentation or respiration, it's got to start out with glycolysis or some alternative to that. Okay, so I've got several different graphics to show you that illustrate glycolysis. Some of them do, will show one thing a little bit better than another. So I like to see it several different ways. So this one is not from your book. First thing to point out is that glycolysis is almost all of life does glycolysis. So it's very highly conserved. It's so popular that we have two different names for it. So we've got a name, the one that really makes sense describing what's going on. Lysis means the split, and glyco, splitting of sugar. All right, so that perfectly makes sense. We often have another name for a pathway that gives homage to whoever elucidated it in the first place. In this case, their names are Emden, Meyerhoff, and Parnas. Okay, so, or the EMP pathway. Okay, and that's a little bit confusing to have something go by two different names, but you just kind of got to recognize both of them. I read research papers where they talk about Emden-Meyerhoff pathway, and it took me a while to figure out kind of what they were talking about years ago. You really just kind of have to recognize both. They both refer to the same pathway. And again, sometimes we shorten it as the Emden-Meyerhoff. Sometimes Parnas gets credit. Okay. But so almost all of life does glycolysis. There are a few bacteria that are missing one of these enzymes, and they have to do something a little bit different. So we'll talk about that in a minute, but almost everybody does it. And think about where this is taking place in the cell. In bacteria or prokaryotes, this is in the cytoplasm, as well as the eukaryotes are going to be doing glycolysis in the cytoplasm of cells as well. All right, so again, we're starting out with glucose here, and the first kind of stage, energy is invested. So we have an input of a couple of molecules of ATP. There's the cleavage step, where basically you have this fructose 1,6-bisphosphate that's cleaved. It's not cleaved in half. This is not a symmetric molecule. So actually, this is a little bit of a simplification here. The next diagram shows that a little bit better. But usually, the one compound is just changed into the other one. So it's kind of a reasonable simplification. You get two molecules of glyceraldehyde 3-phosphate. And then we get some energy out of it in the form of some electrons and ATP, and end up with two molecules of pyruvic acid, same thing. Okay, so let's make a note here of our net gains. Per one molecule of glucose, we get 2 ATP. Again, we invest 2, we get out 4. So a net gain of 2 ATP, we get 2 NADH and 2 pyruvate. So important takeaways there. So then this is the graphic that they have in your book. Important to note that if you have an old version of the book, 4 or 5, there's a glaring error in this diagram. They make you end up with 4 pyruvic acids instead of only 2. So just note that this is the 6th edition one. I like this graphic because it doesn't worry about, the whole structures of everything, but it does pay attention to the carbons. All right, so we're starting out with 6 carbons. We still got 6 at the end because we got 2 pyruvic, 3 carbon pyruvic acids. And this, in the lysis step here, it notes that, yeah, we have 1 glyceraldehyde 3-phosphate, something that's very similar to that, dihydroxyacetone phosphate. It usually just gets changed into glyceraldehyde 3-phosphate. Okay, so it's not an equal split there. Okay, and then I've got this third diagram of glycolysis because I found a graphic that compares our Embden-Meyerhof pathway to a pathway that only bacteria do, and that's called the Entner-Dodorov. Okay, so again, this is only some bacteria. I put up the net gains here just to compare like glycolysis pathway. They've got some kind of, this must be an older graphic because it's kind of got the old notation of diphosphate instead of biphosphate. And it doesn't, have all these names quite written out, but okay, just kind of for comparison purposes. All right, so this Entner-Dodorov pathway, first of all, kind of sounds like Embden-Meyerhof. Okay, it's again the people that elucidated it, and just I guess by coincidence their names kind of sound like Embden-Meyerhof. So that's a little bit confusing. Again, this is a pathway only some bacteria can do it, and the reason that they do it is because they're actually missing. I think it's this enzyme right here, phosphofructokinase. You don't have to know what enzyme it is, but because they are missing or unable to produce that enzyme, they have to have a little different startup here to glycolysis. We actually end up with very similar products, but you'll see here, right? I mean, the glucose 6-phosphate, that's the same. Have to input some ATP, that's the same. And then we have a little different product here, and ooh, look at this. Instead of NADHs getting out of this, we have a different electron carrier, the NADP, a nicotinamide adenine diphosphate. And remember we mentioned before, when you have the NADP carrier, that's taking those electrons to be used for some biosynthetic pathway. It's not taking them to the electron transport chain. So that's one early difference. We have a little different intermediate here that actually gets split. And right away, we have 1 pyruvate as a result there. But we didn't get the ATP out of that we normally would in glycolysis. But we still get a pyruvate. And then the other product of that split is the glyceraldehyde 3-phosphate, which again is, you should recognize as over here. And then we get the NADH and some ATP out of that glyceraldehyde 3-phosphate. So over here, net gains from the Entner-Dodorov, we're getting 1 NADP. Once again, that doesn't get translated into ATP though. We get 1 NADH, and we put 1 ATP in and get 2 out, so we only get 1 ATP. I could add 1 pyruvate. Sorry, 2 pyruvates. Okay, so kind of similar gains. We're getting half as much ATP. Usually not a problem because organisms that use this pathway are respiring. So this is, not the biggest place where they get ATP anyway. They're getting it at their electron transport chains. What kind of bacteria use this pathway? We used to think, oh, it was only like this, and this bacteria. We thought it was pretty rare. And I put a few of those organisms down here just because if you're in lab, you might eventually recognize some of them. Don't memorize them for lecture, but Pseudomonas, that's a genus that you're going to use in lab. Rhizobium, they're nitrogen fixers. The Enterococcus faecalis, that's a organism that's very similar to Streptococcus. But here's one that you'll recognize, E. coli. Well, we now know that some organisms will do the Entner-Doudoroff pathway instead of glycolysis if you just grow them on a certain kind of media. So if you give them certain substrates, then they'll use a different pathway. Okay, so that's sort of interesting. We just know in certain situations, some organisms may be able to do both, but they will typically only do one at a time. They'll either do one or the other. Again, this is we call this an alternative. Whoops, I was afraid that would happen. Ignore that. Okay, alternative to glycolysis. Okay, so some bacteria can do it. They don't, again, usually do them both at the same time. There is another alternative to glycolysis. Let me write that up here. Okay, and that's called the pentose phosphate pathway. It is also referred to as a hexose monophosphate shunt. I never call it that though. There are a couple other names for this. I will always call it the pentose phosphate pathway. This is a pathway that we do as well as bacteria can do. And we may do this at the same time as we're doing glycolysis. So when would we have sugar that we would funnel through the pentose phosphate pathway rather than glycolysis? When an organism's needs are for biosynthesis instead of for energy. Okay, so organism needs energy, it's going to go through glycolysis. If there's, an abundance of glucose and that's not an issue, then they need to make cellular structures. They're going to need to funnel some of that glucose through some enzymes of the pentose phosphate pathway. So this actually starts out a little bit more similar to the Entner-Doudoroff pathway. Right away we have an input of ATP, and right away we get some electrons picked up by NADP. Okay, so we'll see here it says 2 anabolic reactions. Okay, so the electrons, and I'll just write that again, electrons used to power, again, we'll say anabolic reactions. Just make sure you don't miss that. Right here and here, we lose a carbon dioxide. So we call that a decarboxylation. So now we only have 5 carbons to deal with. And this looks very complicated down at the lower end of this pathway. Basically, the organism is not making all those different, or the pathway is not making all those different products. It depends on what the cell needs as to exactly what gets made. Okay, so if the cell needs to synthesize nucleotides, all right, then it's going to utilize this the pentose sugar, 5 carbon, ribose 5 phosphate. Okay, so ribose, those are pentose sugars, 5 carbon sugars. Here's some building blocks to make nucleotides. Okay, let's see, so key pentose sugars for nucleic acids. Okay, the other product that can come of pentose phosphate pathway for bacteria is this erythrose 4-phosphate. Okay, so here it says to the synthesis of amino acids. Let's just say key intermediate to make amino acids, AA. And it's actually aromatic amino acids. We cannot synthesize aromatic. Those are amino acids that have a ring structure. Humans cannot do that. Some bacteria can. Okay, we'll say the rings or aromatic amino acids. Okay, so again, depending on what the cell needs, it will kind of cycle through there. You may have some leftover carbons. You might have some glyceraldehyde 3-phosphate left over, and that can shuttle, back into, whatever step of glycolysis, kind of the leftover bits from the pentose phosphate cycle. Okay, so it's important. There's no ATP produced directly from the pentose phosphate pathway. You may actually end up getting some out of it, again, if some gets shuttled into some pathway of glycolysis or Entner-Doudoroff. Okay. And your book I think your book really only talks about pentose phosphate and Entner-Doudoroff very briefly. I think your book says that like 1 or 2 ATP are generated, but again, that's only depending on whatever is left over there. Okay. Okay. So whatever form of glycolysis is taking place there, typically we're going to end up with some 3-carbon pyruvic acid. And to enter into the next stage of the respiration pathway, the Krebs cycle will only can only work with 2-carbon molecules. So there has to be a little bit of change to that pyruvic acid so it can be funneled into the Krebs cycle. So we call this the transition step. That's kind of like right here on our diagram there. So we have to lose a carbon. That's accomplished by decarboxylation. So we lose a carbon to carbon dioxide. Now we have our 2-carbon acetate that combines with a coenzyme. And in that process, we get a little bit of reducing power here via ending up with our product here, acetyl coenzyme A, which is ready again to be shunted into the Krebs cycle. So what do we gain from this transition step? And you can always count this per pyruvate or per glucose. Remember, we got 2 pyruvates from each glucose, so you just have to make sure it's clear how you're counting it. We'll say per pyruvate, you're going to gain 1 NADH and 1 acetyl-CoA. Okay, it would be 2 per glucose, 2 NADH or 2 acetyl-CoAs per glucose. All right, so then that molecule is going to enter into the Krebs cycle. Okay, another one of those processes that we have several different names for. I usually call it the Krebs cycle because it's just the easiest thing to say. But this is also known as the citric acid cycle, because as you'll see here, whoops, that doesn't spell anything, cycle. As you'll see here, the acetyl-CoA combines with this 4-carbon oxalic acid to make citric acid, 6-carbon citric acid. Okay, so that's where the name citric acid cycle comes from. That's sort of the first substrate of it. It's also known as the carboxylic acid. Actually, tricarboxylic acid. Also known as the TCA cycle. Sometimes we call it that because there's lots of these intermediates that actually have 3 carboxyl groups. That's COOH, that's a carboxyl group. So, and then Hans Krebs, he's the one that elucidated it. So again, 3 different names. all of them refer to the same thing. Okay, and once again, we're not going to get hung up. We don't need to know all the intermediates here, for my purposes anyway. If we think about what we're gaining from the Krebs cycle, oh, and also remember this is in the cytoplasm of prokaryotes, and it's in the matrix of mitochondria for eukaryotes. Anyways, good to think about where things are happening. All right, so let's talk about net gains, let's say per glucose. And I'm going to count the transition step. Okay, so you always gotta, again, say if you're counting it per pyruvate or per acetyl-CoA or per glucose, and do you count that NADH that you gain in the transition step? So if I did, so that one from the transition step, and 1, 2, 3, 4. So, and then per glucose, that makes it 8. So 8 NADHs gained out of that. So that's really the biggest thing that we're harvesting from this Krebs cycle. Lots and lots of electrons. They're going to, again, get turned into ATP. Only 2 ATP. Down here, and remember, that is by substrate level phosphorylation. It's not clear what substrate has a phosphate on it. Apparently there's some phosphate associated with this succinyl-CoA that they don't really show there. Okay, so only 2 ATP by substrate level phosphorylation, 2 FADH2, over here, another carrier. And this isn't really something that we generally care about all that much, but I'll just make a note of it here. We actually get 6 carbon dioxides as a result. So we started with glucose with 6 carbons, and now we've lost all those carbons to the air in carbon dioxide. Some bacteria can't afford to lose that much carbon. They need it to build cell walls in particular. So there is kind of a modified Krebs that's, again, kind of a modification so they don't lose all their carbon. But it's kind of interesting just to think about. All right. It's pretty much all I have to say about the Krebs cycle. If you got any questions, throw it out there. But again, just keeping in mind what we're gaining from it there. Okay, so we are, okay, good. So we are got partway through respiration here. We are ready to take a look at that electron transport chain. We're going to find out what happens to all those electrons that have been gathered by the NADH and some FADH as well. Okay, so the electron transport chain, this is a series of redox reactions that's taking place in the cell membrane. Okay, so let me think. I think I have room here. So in prokaryotes, this is the cell membrane, plasma membrane, same thing. In eukaryotes, again, this is in the mitochondria, the matrix membrane, the mitochondria. Okay, that's mitochondria has one membrane around it, and then it has these cristae in there, like an inner membrane. It takes place in that inner membrane of the matrix. Sorry, so not matrix, it's an inner membrane. That's what I'm trying to say. Inner mitochondrial membrane. The next picture shows that a little bit better. So it's very important that you have an intact membrane because we're actually forming a concentration gradient. There's going to be lots of protons forming here outside or getting pumped outside of the cell, many more than inside of the cell. And I like the edition 6 of the book draws the electron transport chain at kind of an angle down, and that's not an accident, because that's the direction the electrons flow as they lose energy. So, and we can actually measure that. We can put electrodes in the membrane and measure that difference in potential. When the electrons are dropped off at one end, they have a lot of energy, and as they're passed down, they kind of lose energy. So, you put a ball at the top of the hill, it's going to roll down really quickly. If you start it halfway up, it's going to be a little bit slower, right? There's just less energy there. Okay, so we have a series of electron acceptors, well, not acceptors, electron carriers that make up the electron transport chain in the membranes. And I don't need for you to memorize them and what order they are and what they are, because there's a lot of variation between different organisms as to what carriers they use, and even the order that the carriers are in. Even E. coli can have different carriers depending on what media you grow it on. So don't memorize that, but you should sort of understand what kind of molecules are electron carriers. Okay, so here's some examples of the kinds of molecules that make up electron transport chains. Some are considered flavoproteins. That means they contain flavin, which is a coenzyme. Flavin mononucleotide here, that's the first carrier. It just cycles between an oxidized and a reduced state. So back and forth, that's the important thing. Cytochromes, we have several different cytochromes. They all have their own, cytochrome C or B or whatever. Again, they have iron in them that allows them to alternate between being oxidized and reduced. And then ubiquinones, they are just considered non-protein carriers. So coenzyme Q, for example, is considered an ubiquinone. All right, so I would never say, okay, what's the order of the carriers in the electron transport chain? But I may say, what's a ubiquinone? Some multiple choice question to deal with that. You should recognize that oh, that's something important in the electron transport chain. Okay. So as those carriers are passing the electrons, some of them are pumping protons out of the cell. Okay, so we get built up a proton gradient, meaning more protons outside of the cell than there are inside of the cell. And then we refer to that as a proton motive force, meaning it's all this potential energy, kind of like the lake that's held back by the dam. That's all potential energy, because when that water flows through at one spot, it powers the turbines to make electricity. Those protons are going to eventually diffuse down the concentration gradient into the membrane, because they want to go from high concentration to low concentration. And that's going to power the production of ATP. All right, so this is a little bit, it's a little bit confusing, this ATP here. That's not actually where the ATP is produced. It's actually produced at an enzyme complex down here that the next graphic shows better. But this is just showing approximately when enough electrons have been passed to be enough to form an ATP. So when NADH up here is oxidized to NAD, again, that drops it off kind of at the top of the hill. We figure per NADH, we get about 3 ATP. FAD drops its electrons off a little bit lower down the hill. So for each FADH2, we figure on about 2 ATP. Okay, so a little bit less. And of course, there has to be some final electron acceptor. Okay, that can be oxygen. In aerobic respiration, that's oxygen. we're talking about anaerobic respiration, that's going to be something else. Some inorganic ion, maybe nitrate, maybe sulfate, something like that. We'll talk about that a little more shortly here. So it doesn't have to be oxygen when we're talking about bacteria. Of course, we never do anaerobic respiration, but some bacteria can. Okay. Make sure I didn't miss anything. So here's the graphic that they show in your book, which is good in some other ways. Kind of shows you approximately, where it takes place. Here we've got our NADH being oxidized back to NAD. You'll notice all these protons that are building up outside of the cell. And this shows a little bit more nicely the enzyme that actually phosphorylates the ADP to ATP. It's called ATP synthase. Makes sense. So only at those ATP synthase channels, that's the only place that those protons can diffuse back in. And then that powers the production or the phosphorylation of ADP to ATP. This picture also shows oxygen when it accepts those electrons that have been passed down by the carriers. It actually, those electrons complex with a half of an oxygen. So an oxygen molecule has to split to collect those electrons, and then we end up with water. That process doesn't always happen just right. Sometimes those electrons don't distribute themselves properly, and then we can end up with one of those toxic oxygen compounds, superoxide, peroxide, some of those things that we talked about that some bacteria have to have a way to detoxify those toxic oxygen compounds in order to be able to live. Okay, if you get enough superoxide building up, then that's going to be toxic to the cell. In addition, sometimes instead of the electrons being passed from carrier to carrier, sometimes a few of the electrons will leak inside of the cell and again, attach to oxygen in an inappropriate way. Okay, so that's another way that we get superoxide. So cells have to make superoxide dismutase in order to detoxify that. If they can't, then they're not going to be able to live in the presence of those. So an organism might be considered an anaerobe instead. Okay. All right, so a couple, I just gave myself a little bit more room to write if I needed it there. A couple of terms that are fairly confusing that I want to make sure we use in a correct way: chemiosmosis and proton motive force. They kind of sound like they're kind of the same thing, and they are very closely related. So we talked about, we get all this proton gradient, protons outside of the cell. So that potential energy we refer to as a proton motive force because of that buildup of protons. Once those protons are allowed to diffuse in through that enzyme complex, ATP synthase, we call that process chemiosmosis. All right, so kind of the diffusion of protons through the membrane, and that's the process that generates ATP. It's because of the proton motive force, okay? If there wasn't that gradient buildup, then the chemiosmosis obviously couldn't take place. We wouldn't have a concentration gradient. So just trying to use those terms correctly, you see them both everywhere. All right, so if we try to kind of summarize everything, so total output from aerobic respiration, we can do this in kind of a theoretical way. So for prokaryotes, if we add this all up down here, assuming 3 ATP per NADH and 2 per FADH2, then we come up theoretically with 38 ATP per glucose. Now, in reality, that's probably never achieved because bacteria sometimes will use their proton motive force for other things. They'll use it to transport other molecules into the cell, and then they don't have to use ATP. Bacteria use a proton motive force to generate their flagella movement. All right, so whether that, all of those electrons actually get generated into ATP, probably not in most cases. For eukaryotes, theoretically, a little less, 36 ATP per glucose, because they spend a little bit of ATP transporting the NADH into the mitochondria from glycolysis. So remember, glycolysis is in the cytoplasm, and then to transfer that into the mitochondria where the electron transport chain is, you have to actually spend a little ATP to do that. So the theoretical output is a little bit different. Okay, so in the time that we have left here, I know that we're almost used all our time up. I said, what if there is something other than glucose available to the cell? Like I said, they're probably not going to choose something like a protein first, but in bacteria, remember, they can't just engulf some macromolecule because they have a nice rigid cell wall. They have to secrete some kind of enzyme extracellularly. So like a protease is an enzyme that chops up a protein. And remember what proteins are made of? Amino acids. All right, so it's chopping up this protein into these amino acids. The amino acids can easily be transported into the cell. Typically, there's going to be a deamination reaction. So if you're in lab this week, one of the enzymes that we're testing for is phenylalanine deaminase. Okay, that's an enzyme that bacteria would need if they're going to use phenylalanine as a nutrient source. Okay, and a deaminase just means that they cut off this amino group from whatever amino acid it is. And then what do you get left with? There is a 2-carbon compound that can be shunted right into the Krebs cycle. Okay, so that's typically where a protein would get entered in, and then, right, you get the electrons from the Krebs cycle, a little bit of ATP. But the other reason I bring this up, that amino group, that gets changed into NH3, which, what is NH3? It's ammonia, right? And when you see ammonia, you think alkaline, okay? So we get alkaline end products whenever organisms metabolize proteins. So that's kind of nice. In the lab, we throw a pH indicator in the tube and we can tell, if, hey, if we have acidic end products or alkaline end products, that gives us a hint as to what's going on, what is the nutrient is being metabolized. Okay, typically when sugars are used, organisms are going to have acidic end products. Because they're almost doing some fermentation, we always get acidic end products from fermentation. Respiration end products are going to be a little acidic as well. Okay, and so that's just kind of a little, it's more complicated than that, but that's kind of a beginning of protein catabolism. What if lipids were being catabolized? In a normal semester, we test for this enzyme lipase in lab as well. That's one of the things we had to skip this semester. But there's an enormous amount of energy that's able to be extracted from lipids. Lipids, of course, are made up of fatty acids and glycerol. So we've got this 3-carbon glycerol in their fatty acid chains. That's what a lipid is. So if an organism is going to be able to utilize a lipid for energy, it's first got to separate the glycerol from the fatty acids. It does that with lipase. And then the glycerol, even that little 3-carbon compound, that gets changed into dihydroxyacetone phosphate. That was when we split the splitting step in glycolysis, that was the one thing that usually gets changed into the glyceraldehyde 3-phosphate, that unequal split. So that gets shunted right into glycolysis, easy enough. And then the fatty acids are broken down by a process called beta oxidation. Beta referring to the next to the last carbon in the chain. So basically, those fatty acids are cleaved off in 2 carbon bits. There's some electrons gained just by that cleavage of acetyl coenzyme A with those 2 carbon acetyl groups. And then each one of those acetyl groups gets entered into the Krebs cycle. So we get even more electrons from that. So an enormous amount of electrons gained from that beta oxidation. That's a process by which the fatty acids are catabolized, basically. Okay. So we will pick out, that was a lot of pathways to be running through. A week from today, we'll pick up with anaerobic respiration and fermentation and start our next unit in genetics. So see me if you have questions. Office hours all day tomorrow. Email me any questions, and we'll see you Wednesday.